In order to get the insights of the gene function, it is important to evaluate them at the level of messenger RNA, that how much messenger RNA it is producing. But there is one of the limitation if we are going to evaluate the gene function by measuring the quantity of messenger RNA, because the messenger RNA, it is going to convert into protein, and this is the protein which is the actual functional product of any gene. So the study of messenger RNA, one can say that it is just a pre-gene study or pre-protein study, because if the messenger RNA, they are abundant, it is not necessary that all the abundant messenger RNA copies, they are going to convert into corresponding proteins, because after transcription there is gene regulation or post-transcriptional regulation that is also very important, because if messenger RNA has been synthesized, it is not necessary that all messenger RNAs they will be translated into proteins. कि ये ज़रूरी नहीं है कि सारे के सारे messenger RNA जो कि सेल के अंदर हैं वो प्रोटीन बनाएं, या ऐसा भी हो सकता है कि उनका जो ट्रांसलेशन का जो रेट है वो वेरिएबल हो सकता है. And an additional possibility is that messenger RNA, it is not converting into protein at all. So this factor, it is very important. That is, what is the rate of conversion of transfer rather than messenger RNA into protein, and whether the RNA, it is going to convert into protein or not. जैसा कि मैंने बताया कि कुछ RNA जो हैं वो प्रोटीन प्रोडक्ट में कन्वर्ट ही नहीं होते. So this is one of the factor. Another factor is that after translation, when proteins have been synthesized, even then the diversity of the protein, it may be variable. अब प्रोटीन क्यों डाइवर्सिफाइड हो जाती हैं अपनी सिंथेसिस के बाद, तो इसमें इसकी जो पोस्ट ट्रांसलेशनल मॉडिफिकेशंस हैं ये बड़ा रोल प्ले करती हैं. कि जब प्रोटीन का मॉलिक्यूल सिंथेसाइज होता है अकॉर्डिंग टू द मैसेज ऑफ द मैसेंजर आरएनए, तो इसमें डिफरेंट फंक्शनल ग्रुप्स इंट्रोड्यूस करवाए जाते हैं. दैट कैन बी डिस्कस्ड under the heading post-translational modifications of the proteins, and those modification may include like phosphorylation or acetylation, glycosylation, and many other modifications are there that can also accommodate the diversity of the proteins after translation. As I told you that actually the functional proteins, they are important, because if genes are there, their action, their function can be studied in terms of the biological activity of the proteins. Under some circumstances, if protein it is being synthesized, even then it is not functional, although it is present within the system. So under such circumstances, its biological activity, it is evaluated. Another possibility is that the localization. If protein is functional, it is not localized somewhere, compartmentalization is not there. If it is not trafficked properly at its particular location, even then it will not be functional. For example, in case of bacteria, some proteins that are synthesized within the cells, they may be transported into the periplasmic space, that is space between the outer and inner membrane, where they will become functional. So under such situation, the localization for the function of protein, it is also very important. So in order to evaluate the function of the protein, different techniques we must apply, and all these techniques that can study the function as well as can evaluate the structure of the proteins, they can be discussed under the heading that is proteomics, and this field was introduced in late 1997 maybe, and it is a term that was introduced in comparison with the genomics. So if we define a proteome, we can say that as far as proteome is concerned, it is the total set of proteins that are synthesized by a given cell or organism. It may be called as the proteome, and its analysis, it may be called as the proteomics. Now proteins, they also contain very important data that can be studied, although they can be studied like RNA, like their sequence can be studied, their three-dimensional structure can be studied. But in addition to that, some other point must also be taken into account while studying that what is their three-dimensional structure, compartmentalization or localization, what is their physiological or biochemical function, and how proteins they are interacting with other molecules or proteins.